I would like to welcome everyone at RotaChrom's webinar. I am Anita Kis, Senior Research Scientist, originally a synthetic organic chemist, and I'm the responsible one for chiral separations at the company. The presentation will take approximately for 30 minutes, and after that there is going to be an open Q&A session. If you have any questions, you can ask directly in the chat function or at the end of the presentation by raising your hand. The webinar is going to be recorded and it's going to be uploaded to our webpage where you can access it in the future. So this webinar's topic is about the separation, purification and isolation of different types of isomers applying centrifugal partition chromatography. Let me introduce our agenda for today. First, I'm going to talk about the increasing awareness of the application of a single isomer, then about which techniques can be deployed in a problematic mixture, and why CPC could be a strong candidate to solve these purification issues. Then I'm going to introduce you briefly to the working principles of CPC, and after that we continue with guiding you through 4 different case studies. These studies cover the purification of the 3 most important types of isomeric mixtures, most problematic ones. And the content of this webinar can be found in more details in our latest brochure. The cover is visible here. The title is Isomer Separation. It has been released in October, and you can find it via this link here or by scanning the QR code. So let's start. Over the past 2 decades, there is an increasing awareness of the benefits of the application of single isomers in almost every industrial field. To resolve this problem, conventional methods like crystallization or distillation are many times not enough powerful, as they rely on differences in physical properties like boiling or melting points. When such disparities are minimal or non-existent, these methods become ineffective. Moreover, a compound may have several structural or stereoisomers, or even both sometimes, that can share very similar or identical physical and chemical properties, making separation even more complex. Despite this, differences in partition coefficient values or solubility still can provide an advantage for liquid-liquid chromatography applications. Why the application of a single isomer is more advantageous than using a mixture? First, the administered dosage can be lowered, pharmacokinetics and dynamics can be improved, structure activity and dose response relationships can be more understood and reconsidered, of course. And last but not least, it's very important, possible toxicity can be eliminated. There are 3 different synthetic developments that can be reconsidered. First, using enantiomer starting materials for the synthesis, or a targeted asymmetric synthetic pathway for the production, which could require, of course, transition metal catalyst. Both types can be rather expensive on large-scale operations. And last but not least, employing different purification techniques for intermediates or directly for the product, of course. Anyhow, there is a need for further development in separation science because new challenges arise from day to day, and isomeric purity is strongly involved everywhere. I guess this scheme can be very familiar for many of us, but please let me brush it up a little bit. Isomerism, where 2 or more compounds have the same molecular formula, but they differ in connectivity. Those are called constitutional isomers. Or when they differ in spatial arrangements, those are called stereoisomers. And these may result in different physical, chemical, or biological properties. Constitutional isomers are usually quite easy to separate from each other. Mostly separation occurs by their differing polarity, while stereoisomers can be trickier to deal with. Conformational isomers, these ones, are usually less important for separation science. They simply can transform into each other by rotating around their sigma bonds. In contrast to that, configurational isomers can be interconverted. Geometric isomers, these ones more well known as cis-trans isomers, can't rotate around their pi bonds. Optical isomers, these two types. Optical isomerism occurs when their atoms are differently oriented in space. This property is often due to the presence of an asymmetric center that is not always a carbon, and that is called chiral center. To take it simple, when the molecules are mirror images of each other, like these two here, we call that enantiomeric pairs, and when there is no such relation in between, like these two or these two, they are called diastereomers. The physical properties of the enantiomers are totally identical. They only differ in the direction in which they rotate the plane polarized light, and in every other respect, they are chemically identical in a chiral environment. So separation of an enantiomeric pair is the most problematic of all. For the isolation of enantiomers by chromatography, the application of a chiral selector is needed. The case studies included today are going to be presented in order of difficulty of the separation problem. Purity matters the most in these industrial fields, where usually HPLC is the most often applied technique, but SFC has also started to gain popularity during the last decades. And capillary electrophoresis is popular as well, but mainly in academia since it's limited to analytical purposes. For enantiomer separations, chiral selectors are needed. Here are the most popular ones indicated. I'd like to highlight that cyclic oligosaccharides, like cyclodextrins, are the most important. Their remarkable solubility makes them suitable selector for liquid-liquid chromatographic purposes. And furthermore, they are rather cheap in bulk and they can be easily recycled, that I will talk about later. Huge variety of different CDs are available, and having different physicochemical properties make them widely applicable. They can simply act as co-solubilizer for the sample to be injected, or as a chiral selector in one phase of the biphasic system. I've tried to summarize the pros and cons of applying HPLC or SFC or CPC in this table here. HPLC, the name speaks for itself, offers high resolution and theoretical plate, although it can be expensive and the loading capacity is quite limited too. SFC quickly delivers high efficiency and resolution, but the biggest issue can be the low polarity of the applied CO2 as mobile phase. For both techniques, columns are interchangeable but rather expensive, and they tend to degrade quite fast. In contrast, CPC is a technique where there is no solid stationary phase applied, thus its fouling can't happen. Dealing with only liquids allows high loadability, of course, and that is strongly depending on the applied solvent system. Despite offering lower theoretical plate than HPLC, the careful selection of the solvents can enhance resolution. So what is CPC exactly? Where does it belong among other purification techniques? Let me introduce it to you. So chromatography is usually applied when other purification techniques tend to fail. CPC is a hidden gem in the family of chromatography, a purification technique amongst the well-known more traditional methods like extraction, distillation, crystallization, and so on. For enantiomer separation, crystallization, and chromatography are the most often applied techniques. Let me specify what is exactly liquid-liquid chromatography first. So the driving force of the separation can be based on these three forces: gravitational, electrical, and intermolecular forces. Those can be cohesion or adhesion. For CPC, gravity is that force, what is driving the separation, but more specifically, it's because of density difference. The geometry of the space where separation takes place can be planar, like thin layer chromatography. It can be a column like HPLC or flash, or it can be a rotor like CPC. There can be either physicochemical or biospecific interactions between the solute and the stationary phase. Most often it's physicochemical, and CPC is a preparative technique. And the last category is defined by the state of matter of the applied phases. When the stationary phase is solid and the mobile phase is liquid, it's called LC. When there is a solid stationary phase and the gas mobile phase, that's GC. And when the mobile phase is in supercritical state, it's SFC. And when both phases are liquids, that's LLC. So centrifugal partition chromatography is a type of liquid-liquid chromatography. For a separation by CPC, a biphasic liquid-liquid solvent system is needed. It can be binary, ternary, or even quaternary, and by their density difference, separation occurs. Molecules partition coefficient determines its concentration in the two phases of an immiscible solvent system. Measuring it can be done by this shake flask method visible here in the animation. A mixture containing different compounds with different KDs goes in the tube, flask is shaken, and each compound with different KDs distributes in different amount in the two phases. You can see it on the chromatogram. When a compound's KD is 1, it has been equally distributed in both phases. So see the yellow dots here. And the peak comes in the middle of the spectra with a nice Gaussian shape. And when the KD is smaller than 1, then smaller than 1, the compound is more concentrated in the lower phase, is the yellow dots now here. And the peak is coming before the one with the KD1 on the chromatogram. And it's also sharper and with smaller full width at half maximum here. Instrumentally, in CPC, there is a rotor instead of a column, and where actually the separation takes place. Numerous cells are attached to the rotor, which are interconnected by series of ducts, these ones. And by making the rotor rotate, we create centrifugal force. And by this, one of the phase is immobilized in the rotor, providing stationary phase. The mobile phase containing the sample to be purified is fed into the rotor and pumped through the stationary phase in the form of tiny droplets. There are two fundamental operation modes exist. When the stationary phase is the denser fluid and the mobile phase is lighter, arriving into the cell against the centrifugal force, so upstream, it's called ascending mode. When the stationary phase is the lighter fluid and the mobile phase is the denser one, entering the cell in the same direction as the centrifugal force, we call that descending. Other operation modes are possible to carry out in CPC, but we don't include them in this webinar. So quickly, I'd like to introduce you our product portfolio. RotaChrom's smallest benchtop size instrument is the CPC modeler with 250 milliliters of rotor volume for smaller laboratory scale separations. A pilot scale version of it is available. It's called the RCPC, this one, with 2.2 liters rotor volume, and chronologically our first instrument was the ICPC, which is our biggest one. Industrial scale equipment with the rotor volume 23 liters, and by connecting two RCPCs on a specific way, continuous operation mode is achievable. That is our newest product in line. What is really important to mention here that the applied method scalability from smaller to bigger instrument is completely linear with CPC. RotaChrom's instrument can handle increasing levels of demand complexity and measurement range without compromising performance. What you can achieve in small scale, you get exactly the same on industrial scale without losing accuracy or efficiency. So let's continue with the case studies. All of these projects were carried out for a request of industrial collaborators. The projects are discussed here in order of difficulty of the separation problem. So the first one is quite easy. It's isolation of different constitutional isomers of vitamin E. Palm oil is an edible vegetable oil. It is widely applied nowadays in the food industry, cosmetics, and biodiesel production. In 2023, 77.5 million tons of palm oil was produced worldwide. Due to ecological reasons, it is the most sustainable and acknowledged plant among the others in terms of land and water usage. The overall goal for this project was first the enrichment of tocotrienols from raw palm oil, and then their isolation from one each other. The crude material contained 37% of the targeted tocotrienols along with 12% of tocopherols. Tocopherols and tocotrienols are the isomers of vitamin E. Structurally, all the isomers of vitamin E share the same heterocyclic chroman head, benzodihydropyranol head, serving, of course, hydrophilic functions. They differ only in their isoprenoid side chain on C2 carbon here. They act as, of course, lipophilic auxiliary. Tocopherols have a saturated phenyl tail, while tocotrienols bear an unsaturated chain with 3 double bonds in transposition. Moreover, they differ in the substitution pattern of the methyl groups on the chroman head. So vitamin E consists of 8 constitutional isomers, 4 tocopherols and 4 tocotrienols, namely alpha, beta, gamma, and delta isomers. As I mentioned before, it is known that different isomers can show remarkably different therapeutic effects. For example, the differently substituted tocopherols are used as different food additives, while different constitutional isomers of tocotrienols serve in sunscreens as neuroprotective agents. The chosen solvent system for this separation gave us enough selectivity to differentiate tocotrienols with these partition coefficient values listed in the table here. So having a suitable solvent system in hand, carried out the isolation on our lab scale instrument. Separation details can be found in this table here. The run was carried out in descending mode. On the chromatogram, we can see successful separation was carried out by the applied method. In elution order, delta, gamma, and alpha tocotrienol were almost baseline separated with very good selectivity, and they are also completely separated from the rest of the tocopherols here. We achieved 90% of purity for tocotrienol content and 97% isolated yield. The next case study covers the separation of a diastereomeric pair in the precursor of the nebivolol. It was a little bit trickier project than separating constitutional isomers. Nebivolol is a commonly prescribed selective beta blocker and antihypertensive medication. Yearly, more than 3 million prescriptions are handled only in the US. One of its key symptom equivalent is this chroman epoxide being formed as a diastereomeric pair in one of the earlier steps of the multi-step synthesis. Nebivolol is having 4 asymmetric carbons, which would allow the formation of 16 stereoisomers at the end of the multi-step synthesis. unless there is a highly stereo-controlled synthetic pathway available for the formation of the desired stereoisomer with higher diastereomeric access, or if there is any targeted purification for one of its intermediates. The goal of this project was the separation of the chroman epoxide diastereomeric pair. The diastereomeric ratio was 3:6 for A and B diastereomers. A ternary solvent system obtained 1.5 and 0.8 partition coefficient values, respectively for A and B isomers, by this achieving selectivity factor as high as 1.87, in descending mode. And please keep this in mind, this is in descending mode. So we have carried out the separation on lab scale again first. The details here in the table, although I have to highlight that the chromatogram was carried out in ascending mode, so not descending mode, and the PIX elution changed. So the diastereomer B comes first in ascending mode. And because we did it because it made the following evaporation much easier, since both compounds were received in the upper organic phase. Otherwise, descending mode would have resulted to obtain the diastereomers in the water phase. And everybody knows, I guess, here that it's not really nice to evaporate something from water. So baseline separation occurred. We obtained 99 plus purity for both A and B diastereomers, above 95% isolated yields for both products. One more diastereomer separation, sterically more demanding than the previous one was. It was about to isolate amber kettle for perfumery purposes. Amber kettle is an odorant, one of the few synthetic equivalents of naturally occurring, highly valued ambergris. The molecule itself is naturally abundant in the bark of the western white pine tree. Its sustainable synthetic production is gaining importance nowadays due to environmental concerns and restrictions. Structurally, amber kettle here is a polycyclic labdane type terpenoid having 5 asymmetric carbons indicated on the structure here, which would allow the formation of numerous stereoisomers. But by the industrially applied synthetic conditions, these two form only. It has also been reported that while amber kettle has very strong woody fragrance, iso-amber kettle is odorless totally. Throughout the whole multi-step synthesis, racemic polycyclic intermediates are generated. Thus, a suitable purification method is strongly needed. Previously, neither column chromatography nor fractional distillation proved to be cost-effective for this separation. A ternary solvent system was found to result in 2.4 and almost 4 partition coefficient values, delivering 1.65 selectivity factor. So the separation was carried out on the lab scale instrument. Again, details are here. And the separation, the RAM was in descending mode, elution mode, and it resulted in 85% isolated yield and more than 95% pure amber kettle. For the request of our partner, we scaled the method up to our pilot scale instrument, where we obtained this same decent separation with fairly good 30 gram per hour throughput. Now the stakes are getting quite high here with the separation of the enantiomers of voriconazole. Voriconazole is a widely used third-generation antifungal azole type drug. The molecule has 2 asymmetric carbons, allowing to form 4 stereoisomers and enantiomers of 2 diastereomeric pairs. The preferred synthesis of voriconazole leads to the formation of a racemic mixture of the 2R3S and the 2S3R enantiomers, where this one is the Isomer, the active pharmaceutical ingredient, and this one is the isomer. Industrially, optically pure voriconazole is obtained by resolution, and resolution, to be honest, in most cases tend to be rather a witchcraft than actual preparative preparative chemistry. For enantiomer separations, the application of a chiral selector, as I mentioned before, is usually needed. We found that in a ternary solvent system containing sulfobutyl ether beta cyclodextrin as chiral selector results in selectivity as high as 304, providing 0.5 and 1.3 partition coefficient values, respectively, for the Isomer and the isomer. By seeing these results after the HPLC analysis of the sample, we were quite sure about how nicely baseline separated peaks would appear on the chromatogram. So we have tested the system on our lab scale instrument first, where we achieved 99 plus purity with 97% isolated yield in ascending mode and in a not much more than 20 minutes here. To achieve the best purity with the excellent yield and the highest throughput is kind of idealistic, and most often we can never have it all in the same time. And this study proved this theory. So we've got enormous selectivity, 3.0, excellent purity, 99 plus, and good isolated yields. But in this case, voricanazole's rather low solubility in the applied biphasic system was a drawback. And with the applied batch method, we were not able to achieve higher throughput. So we had to come up with an idea that is the so-called stacked injection method, where the rotor is filled up with the stationary phase and the cyclodextrin is dissolved in it. And it is maintained in the rotor throughout the whole elution. So then injection takes place, and with a specific type of elution mode, we managed to inject 12 times to the same stationary phase without losing the system's selectivity. By this method, 99 plus pure voriconazole obtained with 93% isolated yield, which I would say quite good for enantiomer separation. Please note that voriconazole was received in the upper organic phase, so only evaporation follows as workup, and it first met with the selector on the rotor, so we didn't need to remove that either from the solution. I'd like to also highlight that resolution provides only 37% yield, and usually within days, because hydrolysis is also a necessity, of course. With our developed method, there is a threefold increase in productivity, and the costs are comparable, not to mention that we can save 75% of our stationary phase, which is really expensive. And we can even recycle it. So time is short, and let me conclude. All in all, isomer separation is a challenging process, and most often chromatography is unavoidable. Purity, yield, and throughput forms a magic triangle when, if you are lucky, you can deliver two factors, but there is always going to be a problematic third one, what you will need to play around with. To balance on between these lines is sometimes really difficult. For isomer, separation's purity is a must-have property, the most important one. CPC proves to be able to deliver high purity material, and even if the task is very difficult, like enantiomer separations, excellent to good yields can be achieved. The third factor is throughput, which is quite difficult to deal with, since most often it goes beyond our control, but with the so-called good CPC practice, it can be controlled. To increase throughput and in the same time, of course, productivity, the application of more advanced methods are necessary, like the mentioned stacked injection one, or thinking in continuous solutions or dual rotor working modes. And lastly, it is important to mention that the CPC in the same time offers an economically conscious solution for purification problems too. And thank you for your attention. I don't know if we've got any questions during the presentation, but if you do have any questions, please raise your hand.